Okay, let's look at this retake exam and figure out where you went wrong or where you went right. As you saw from the previous exam, the first question really didn't change much. In fact, most of these questions didn't change that much. It says this scientist isolates a number of non-photosynthetic eukaryotes. In the previous question, it was non-photosynthetic prokaryotes. Which structure would not be found in these cells? And in non-photosynthetic eukaryotes, you would not expect to see chloroplasts. So at this point, if you're getting this wrong, you got to ask yourself: is it because you didn't see the knot here? Is it because you didn't recognize this as being, not a plant, right? Because non-photosynthetic eukaryotes would have membrane-bound organelles, and therefore the chloroplast would not be necessary. Why isn't it cell walls? Because some protozoans have cell walls, right? Why is it that it's not Golgi body? Because you would expect to see a Golgi body in a eukaryote, in all eukaryotes. Why is it not a nucleus? Because you would expect to see a nucleus in a eukaryote. Here we have the same exact graph, and just like in the last, in the original test, the graph didn't change at all. You have L here, and just like I said in the last explanation, the last test, this is just about being able to read a graph and know what's going on in photosynthesis. You need to do both, right? So if you're looking at the rate of photosynthesis and you know that photosynthesis means you're taking CO2 and it means you're taking CO2 and water and you're making oxygen and sugar, right? So CO2 gas is out here and water, right? Or H2O. And on this side, after you're done with photosynthesis, you have sugar and you have oxygen gas, right? Where would you expect to see most of your CO2? Over here before you start doing photosynthesis. So the L region is where you'd expect to see the most CO2. Where would you expect to see the most oxygen? Well, at the end, right? So if everything else stays the same, then oxygen you'd expect to see at the end more than you would at the beginning. Sugar, where would you expect to see more sugar? Again, at the end. And where would you expect to see more water at the beginning? So this is photosynthesis, the rate at which you take this and change it to this, right? So which statement is supported by the data in the graph? The rate of oxygen production is the same in all sections? No. As you said in the previous explanation, and again, I explained each of these before you took this second attempt. I explained it in class and then again in the video. The rate of oxygen production is the same in all sections? No, because you'd expect there to be a flat line if that were true. The rate of oxygen production is lowest in section M. That's where it's highest. It's exactly the same explanation as in the first exam. And the production of water molecules is greatest in section L. Water is not produced by photosynthesis, so that's not an option either. Again, the same exact answer, same exact explanation. Now, this is a little different, and it says the number of carbon dioxide molecules in the beaker is greatest in section L. That's true, right? So that's where this is the answer that's actually true, and that's that carbon dioxide molecules are greater in the section L than they would be in any of the other sections of the graph. If you know what's happening in photosynthesis, and you know how to read the graph, then it shouldn't be a problem. Question 3. Those are fundamentals, right? What's happening in photosynthesis, and how do I read this graph that's going up and then coming down? What's happening at each of the stages in the graph? Which statement best explains the effect of temperature on the rate of photosynthesis? Here again, what's happening on what's happening in the graph? It starts low, goes high, comes down, and drops quickly. Right? That's not even that accurate because it's a little different. It starts low, goes up, and then drops down dramatically. Where this was about 25 degrees here, Celsius, right? After about, I think 30 was somewhere around here, and 40 was around here somewhere, and what you saw is a section L was here, and M was up here, right? So something like that, and you had the advantage of being able to look at that graph. Unfortunately, I don't want to copy and paste it, but you get the idea. Which statement best explains the effect of temperature on the rate of photosynthesis? The rate of photosynthesis is greatest at 30 degrees. Well, no, the rate of photosynthesis was greatest at 25 degrees, the peak of the graph. So this is not correct. The rate of photosynthesis is unaffected by temperature of the water? No, because if that were true, then I would expect to see a flat line on the graph, not a peak and a valley. If a peak and valley means that there's change, right? The number of chloroplasts in a cell increases as temperature increases. No, again, if that were true, then the process would just keep increasing. There would be no increase and then decrease. The enzymes involved with plant photosynthesis are denatured at temperatures above 25 degrees Celsius. That seems the most reasonable of all these particular choices, right? So this is the answer, and it's pretty much the same answer as it was in the previous exam, where I spent time in class explaining it to you, and I gave you that previous video. All right, let's go to the next one. Question 4. Okay, so there was a numbering issue. This should have been 4 and 5. And then it says you have this diagram going on, and then it's a time-lapse diagram, and it shows you circle all statements that apply. So that lets you know that there's probably more than one answer, right? So let's keep that in mind as we move on. So it says the particles crossing the membrane have a charge. Well, let's look and see what's going on here. They get this membrane protein, and it's allowing these particles to go through. These particles cannot go through without that membrane protein. They can't get through here, 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 or here. They can only get through this membrane protein. They're going from high to low, so they don't need any extra energy. They are getting help. So this would be called facilitated diffusion, right? So it's getting help to diffuse because it's going from high to low. If it were going from low to high, that'd be another question. So particles crossing the membrane have a charge. That's possible. So that would be one of the answers that it could be, because if the particles had a charge, they couldn't get through without help. Remember, the only difference between this question here and the question I gave you before, or one of the only differences, is that this picture had no membrane proteins and it had particles on one side that could get through. And if they could get through, they could not have a charge, right? Well, this one, they can't get through. They cannot get through. They need help. So they could be that they're charged. Remember, see this. This says, the question says statements that could describe the particles, and A is one of those could. All right. B, the movement across the membrane requires energy from ATP. Well, that's not true. As I said, it's going from high to low, so it doesn't need ATP. That's the only requirement for the use of ATP to cross the membrane. Easy going from high to low. Again, I've said this 100 times. I've asked you to look at the problem sets. I've asked you to look at the videos. You've done 3 different versions of this same problem in a formal assessments. This is your third time doing it. So the movement across the membrane requires energy from ADH. I just said it doesn't require any extra energy, right? Extra energy, because this is just anything moving from high to low does not require any extra energy. So that's not a problem. It's only when you're going from low to high that you have an issue. The movement across the membrane does not require added energy. Does not require added energy. Well, I just said that, right? So that one's correct. So the answer there is A and D. The particles crossing the membrane could not be proteins or carbohydrates. Well, that would be true if these weren't getting any help, right? In the previous problem, the picture was different. They didn't have a membrane protein, so the particles were moving through on their own. Proteins and carbohydrates tend to be large. They tend to be have polar regions, and because of that, they were not going to pass the membrane easily. Some smaller proteins might, but most proteins and carbohydrates are not going to pass through this membrane without help. This protein could be giving small proteins and carbohydrates help to cross the membrane. That is true. So because they could be, this answer is not right, because it says could not be. All right. So this can't be an answer. This is not an answer, because they could be proteins and carbohydrates because they're getting help. The particles crossing the membrane could be oxygen gas. Well, if they were oxygen gas, they wouldn't need a protein, because oxygen gas would just pass right through the membrane without any help from a protein. But these are getting help. So they could be proteins and carbohydrates, but they cannot be oxygen, not because they're not passing through membrane, but because they are passing through a protein. Oxygen gas would not need a protein to pass through. Particles crossing the membrane could have a partial, weak, or positive and negative charge. Sure, there's no reason why not. Maybe there are large particles that are polar, like a sugar. Carbohydrates have positive and negative ends or side chains. That's why they can't pass through the membrane easily. And proteins do help them pass, so that could be. Particles crossing the membrane could be ions. They could be completely charged. Absolutely, that would explain why they need a protein to cross. So it's A and D, and C and D are the two possible answers there. Let's go to this one. Now, this is a little trickier. It's exactly the same problems you had in 6. The only in the previous test, the only difference is the picture's changed, right? And I also changed the question. I asked you the reverse question, right? In the previous question, they gave you a drawn red blood cell, and they drew some triangles and circles, and then they said, you figure out where it's highest and lowest and where the water's going to move. Here, they're showing you where the water is moving, right? In the first one, you see water's going in. The second one, you see water's going in both directions. The third one, water's coming out. So here, and I meant for C not to be that answer, but it turned out it was correct. So that actually this one has 2 right answers, as it says, choose all that apply, right? So here, you have water going in the first one, and if you remember our rule, and it's just all it is an application of our rule, and you can think of it in several different ways, however you want to think of it. But let's use the simplest version of that rule, and that's where water is going to move where the concentration of solute is greatest, right? So in A, here in A, where is the concentration of solute the greatest then? If what I just said is true. If water is going to move to where the concentration of solute is greatest, where is the concentration of solute greatest here? In the solution that it's in or inside the cell? Well, hopefully you say inside the cell. So that makes the water, the solution the cell's in here in A, hypotonic. Hypotonic. Why? Because the word hypotonic, as I've said many times, hypotonic means lower solute concentration, right? lower solute concentration. And what's the solution hypotonic to? Inside the cell. So, because the question is the solution. So the solution would be hypotonic. All right. B, solution C, is hypertonic. Solution C, where's the water moving? It's moving out of the cell. So where is the solute the highest? It's outside the cell. So the solution is hypertonic, and hypertonic means greater. Greater solute concentration. Greater solute concentration. So hypertonic solutions are going to have greater solute concentration than what? Than whatever they're being compared to, in this case inside the cell. And if we look at this cell and we ask ourselves, inside the cell is you see water coming out, water is moving to where the concentration of solute is the greatest. It means the concentration of solute is greatest outside in the solution. So that's why that solution is hypertonic. So that is a correct answer. So C is a correct answer. It's hypertonic, right? So B is an answer there. Solution B is isotonic. There, and you can see that the water is moving in both directions. And remember, water is always moving in both directions. Water is moving in both directions equally here in B. So it is isotonic. Also, you see that the cell is not swelling up the way they are in A, and it's not shriveling up like they are in C. They're staying the same, and that's because B is isotonic. So the answers are B and C. Let's check D just in case. It says protein and sugar from the red blood cell will diffuse into the solution. We know proteins and sugars do not diffuse. They need help at least, and that would be facilitated diffusion. And A was wrong because it's not hypertonic. So at least this would be in C as the answer. 7, which of these is required for photosynthesis? Select all that apply. You need to have carbon dioxide for photosynthesis. This all goes back, all the photosynthesis questions go back to really some basic fundamentals, right? Carbon dioxide, water are taken in with sunlight, and out comes sugar and oxygen. You need to know that. So what's required for photosynthesis? Carbon dioxide, sunlight, and chlorophyll. And you have to know that the pigment chlorophyll is what's doing the photosynthesis. And you don't need oxygen. You produce oxygen. Again, this is, I don't know what to say. It's just the same stuff over and over again. And if you keep getting it wrong, you must not be memorizing, or you're having trouble reading the questions. I'm not sure what's going on, but I can't imagine getting number 7 wrong if you understand what photosynthesis is. And yet a lot of you got it wrong. So I don't understand. I just don't get it. Question B, is a typical animal cell which component is has least amount of water? The cell membrane might have some water in it as the water's crossing the cell membrane, because remember, water does cross the cell membrane. Cytoplasm has the most amount of water. The nucleus has some water in it. A ribosome is just an enzyme. It's just a RNA molecule that's making protein, so that would have the least amount of water, right? It's pretty straightforward. This question here is about the spontaneous generation questions. These are 2 famous experiments done in the 1600s and the 1800s. They were not covered in class. I didn't want to cover them in class because I wanted to give you the opportunity to look at, be given something, an experiment that you have to analyze. And we've done experiments in class. We've analyzed experiments in class. You need to understand that you have to apply this experiment to, the scientific method and be able to identify, what is the goal, what are you able to imply from these results, what's the control group, those kinds of questions. Plus, that life comes from pre-existing life, and this spontaneous generation only happened once. It doesn't happen, and the story that people thought that things that were dead turned into flies. You knew that going into this question. So let's try to apply that, even though you didn't look at the exact experiment. And remember, on the state exam, they will throw experiments at you that you've maybe not seen before. It's impossible to see and go over every possible experiment, every possible question. You have to apply the fundamentals. So here is Francisco Redi's experiment. And it says a wide mouth jar containing, it's open to the air. There's meat, and flies entered and laid eggs, and the eggs hatched into maggots. And that's what happens normally. And people thought at some point that how do you know that the flies are laying the eggs and not that the meat is just turning spontaneously into flies, maggots and then flies? Well, Reddy just did a real simple experiment. He covered the same red meat in a different jar. Here's your control group without the cover. Here's one with a cover. It allowed air to get in and out, so there's not something magical in air, and light could get in and out because maybe somebody would say it might be light that's making the change happen. So no flies entered, but they laid their eggs on the gauze, and they hatched into maggots. Or eggs fell through the gauze and hatched on the meat, so that didn't really work very well. And then he sealed the jar, and there were no flies and maggots. So there was less maggots and flies on this one, and no maggots and flies on the one that was absolutely sealed. And so Reddy pretty much showed that dead things do not turn into flies. And so spontaneous generation, and if you didn't know what spontaneous generation was, then you had a problem, but again, we covered it pretty well in class. So here is their life comes from pre-existing life. That pretty much shows that life does come from pre-existing life. Dead things, life does not come from dead things, right? Zombies aren't real, not in the traditional sense of the word zombie. All right, so Louis Pasteur in 1859 experiment, he took some broth, so some nutrients, right, some food in water, and then he took it, he sanitized it, he boiled it so that there would be nothing alive inside it, and he allowed it to cool, and fresh air came in, and after a while, that broth that had nothing alive in it had living things, had bacteria. Here he covered that opening with some cotton gauze, and that filtered the air so that nothing alive could get in, and the broth stayed sterile. And then he took and he made a flask that had this kind of loop in it. He bent the glass, and you can bend glass if you heat it up, and they removed the air by allowing the air to enter this little bent area. And what he found that the broth was sterile, so air was getting in, but there was liquid in this bent area. There was a trap here, and any bacteria, living things, got trapped in the neck. And so air could come in, but it would get any living things would get trapped in the neck, and so the broth again stayed sterile, showing again that it wasn't that the broth was turning into living things. The broth isn't turning into bacteria. Bacteria landed in there via the air, and they multiplied inside the broth. That multiplication of cells inside the broth, and here the multicellular organisms landing on the dead things, this is where life came from, not spontaneously from non-living things. So let's look at our answer choices. A, life comes from pre-existing life. Yeah, these experiments pretty much show that, right? That all the life that you saw happening here, and to a lesser extent here, all came from life that existed before. all the other experimental groups showed clearly that life did not come from non-life. Life came from inorganic materials. Well, we know that first of all, that's not what the experiment was about. All the materials in here were organic, so this is no good. All right. Cells form through spontaneous generation. Well, that's what this was disproving. That's what it's showing not. Cells are not the basic structural functional unit of life. This didn't really talk about cells much at all. It did show bacteria in the second set of experiments, but really didn't talk about it. So it can't really be D either. So we're back to choosing only one answer, and that's A. All right. All right, let's move on. Plus, we know that D was true. All right. So 10. 10 shows exactly the same experiment that you saw in the previous test. So let's see what's going on here. Student shows us the picture shows a student's experiment with Elodea, a common aquatic plant. Exactly the same setup, exactly the same question. Choose all that apply. What I think was one of the small changes I made. It says in this experiment, most likely to decrease the volume. And this was another thing, decrease. I changed it from increase to decrease. Those were the two changes I made. More than one right answer, possibly. And it went from increase to decrease. All right. If you understood 10 from the last experiment, you should have been able to understand 10 here and answer the question. Use fewer plants. Would that decrease how much oxygen is being produced? Again, what is photosynthesis? Taking in light, taking in water and light, and turning that into, and carbon dioxide, turning that into sugar and oxygen gas. If I use fewer plants, I decrease photosynthesis, so I decrease how much oxygen is being produced. That would be correct. So using fewer plants is the right answer. Replace the beaker with a larger container. How would that help me decrease photosynthesis? It would not. Move the light source further away from the beaker. Yeah, that would decrease it because the less light I have, the less photosynthesis would occur because I need light for photosynthesis. So A and C are correct. And decrease the amount of water, that would not help me. Increase the amount of water would not help me decrease photosynthesis. If anything, it would help it increase, but it would only help it increase if water was a limiting agent, right? In this case, there's plenty of water, so that's not a problem. So no matter what, the increasing amount of water in this particular situation wouldn't change anything, but it certainly wouldn't decrease the amount of photosynthesis. Because I know, and if you didn't answer this correctly, I wonder if you know that water is necessary for photosynthesis. So if I increase the amount of water, I'm not going to decrease photosynthesis. The next picture, some people were confused about. It says, I'm not sure why. It says clearly the hydrogen ions are being pumped. This was a mistake, and I asked you to correct it, right? So this should not have said being pumped. This should have said moving and it should have said high. And this should have said low, and that's pretty much what I changed from the previous test. That's why I forgot to change the wording. So it's hydrogen ions are moving from high to low, and you can see that. You see it says here region of low hydrogen ion concentration. There's more hydrogen ions out here than there are in here. That's why this is positive and this is negative. The hydrogen ions are moving in from high to low, so that automatically tells me it is not active transport. So right away I can cancel out active transport because everything's moving from high to low. Is it passive transport? Passive transport, what that means is that it's moving from high to low without any help. Well, it's using a protein A, so it can't be passive transport. Is it facilitated diffusion? Well, it could be. It is certainly one of those possibilities. It's getting help from a protein, but there's something else going on here. There's two of them. Two of them are getting transported across the membrane at the same time. Two of them are being transported across the membrane at the same time. So at that point, we'd have to say what we're talking about is not facilitated diffusion. It's not just simple facilitated diffusion. It's actually co-transport. One of them is coming across and it's bringing along with it another one. So two of them are coming in at the same time. That's hence the word co. You know, that again, from the movie I showed you in class, from our discussions in class, and from the previous, I think I mentioned in the previous explanation of possible versions of this question, the previous explanation of the exam. All right, here we go. Which of the organisms in the chart could be prokaryotes? Well, let's look at it. Membrane bound organelle. Well, right away, membrane bound organelles, you know it can't be a prokaryote. But on top of that, it has a nucleus. So right away, I know it can't be W. I know it can't be W because W has membrane bound organelles and has a nucleus. X, cell wall DNA, but no membrane bound organelles, no nucleus, possibly. Y has no cell wall, has nucleus and membrane bound organelles. There are some bacteria that don't have cell walls. They're not as common as the ones that do, but let's leave those two alone. Z, membrane bound organelles and nucleus, it can't be Z. So now let's look at our choices. W, we just said it can't be W, so W can't be. X, it could be X, but I got X or Y. One of those two could be. Z, can't be Z. I already crossed that out. W and Z, can't be W and Z. What's the only choice I have left? B. So was this really, what was this testing? You know, I think if you knew what a prokaryote had, that a prokaryote did not have membrane bound organelles, and you knew it didn't have a nucleus, then you knew it was either X and Y. It could have been X or Y. So that much you knew. If you knew that much and you could read this table, then the answer was obvious. It had to be B. 13 didn't change much at all. In fact, I would almost say nothing changed in 13. Cell membrane controls movement across membranes. Following particles are moving from high concentration to low concentration are using a carrier protein. How could you describe the type of movement across a membrane? Well, we're going from high to low, so that's diffusion. It's not simple diffusion. It's not passive transport because we're using a carrier protein. It's getting help. These molecules are getting help, so they're ions of some kind, or they're other particles that can't cross the membrane by themselves. Again, this is the same conversation I've had over and over again. I feel like I'm repeating myself multiple times. So it's not simple osmosis, because osmosis is the movement of water, and secondly, osmosis doesn't require a protein, although some proteins do help at times with water. Active transport, it's not active because it's going from high to low, so that's not a possible answer. It's not simple diffusion because a carrier protein is involved. The answer is facilitated diffusion. Why is it not co-transport? Because only one molecule is moving across at a time. Co is two molecules. All right, let's move forward. 14. Some people overthought this question, but let's think about it. What do we see in this picture? It says look at the cell membrane in the model below, right? So let's look at this model. So there's this thing that's been drawn for you. We got to think about the model. What do we have? We have two things in this particular drawing, right? We have these lipids, these phospholipids, right? This part, these are all phospholipids, this whole thing here, all these things. And then we got something else. These things are stuck in the membrane. These things. I'm coloring in yellow. And we've covered this in class. We've built these. We've talked about these. We've said we folded these. We stuck them on the wall. We said these are proteins, and they're proteins that The reason proteins work well in the membrane is because they can be folded and you can have different regions, and the regions can be hydrophobic and hydrophilic. It's difficult for carbohydrates to be this way because carbohydrates are just polar, right? In other words, they're hydrophilic. They won't fit in the hydrophobic area very well at all. So they're not going to sit in the membrane. There's only two things that you see in this picture: the lipids, the phospholipids, and these proteins. So let's assume that you know that. Now let's go through and try to cancel some of these out. So there's only two things. I know the lipids are one, but that's the majority. So it's asking me the lesser part, right? It's asking me the lesser part, right? So if it's asking me the lesser part, and there's only two things I see, I know it's not a nucleotide because nucleotides in DNA and RNA. I know it's not a lipid because the lipid's the majority part. That leaves me carbohydrates and proteins. Carbohydrates don't sit in the membrane, so it can't be carbohydrates. The only possible answer is protein. Okay, so in this model, now some people were saying, well, carbohydrates are on the outside of the membrane. They said, yes, there's true that you can have glycoproteins and glycolipids, but that's not shown in this model. Stick to the question you're being asked. So the next question looks at these concentrations of water moving, and it's again another experiment that's being thrown at you, and the percentage of mass of each of these cells, the cells particularly are eggs. This part of the question didn't change at all. You see there's the gained water mass here, lost water mass here, gained water mass here. So here it gained water mass, so water went into the egg, so this was hypotonic. This was hypotonic as water went into the egg. You see 100% water. You can't get much more hypotonic than that. And this is lost water, so corn syrup is actually hypertonic because water left the egg, right? And water moves to where the concentration of solute is the greatest. Water is leaving the egg, so the concentration of solute must be higher outside than inside. So it's hypertonic. Okay. Same explanation as the previous question. So the next thing is, based on this experiment, which of the following should be inferred about the cells with semi-permeable membranes? The cells placed in vinegar were placed in a hypertonic solution. No, they gained weight, so water went into the egg. So the solution outside the egg was hypotonic, not hypertonic. The cells placed in corn syrup were placed in a hypertonic solution. Yeah, they lost water. Water went out of the egg. So in the corn syrup, there was more concentration of solute outside the egg. C, cells placed in distilled water were placed into a hypertonic solution. No, they also gained weight, and it's in distilled water, which means zero solutes. And there's certainly more solutes inside the egg than zero, so it's in a hypotonic solution. Cells placed in vinegar placed in an isotonic solution. Isotonic means equal. That is not correct here. All right, so the only possible answer is B. So 16, the diagram below shows a relationship between photosynthesis and cellular respiration and the organelles in which they occur. Well, this is, I've seen this diagram drawn in class. I drew it for you on the board. We went over it. You've seen the exact same diagram on the previous exam. So it really should not be anything new to you. So which statement describes how photosynthesis and cellular respiration are interrelated? Are these two related to one another? Oxygen is being produced during photosynthesis and stored during cellular respiration. It's true that it's being produced, but it's not true that it's being stored. In cellular respiration, we use oxygen to make water, so that's not true. Carbon dioxide and water released by cellular respiration are used in photosynthesis. Does that sound true to you? I hope it does because it's true. So think about it, and it says it right here, carbon dioxide and water. Of course, you have to know that this is cellular respiration and this is photosynthesis. I'm not going to go through that explanation again because I went through it in the previous video. So this is cellular respiration, this is photosynthesis. So water and carbon dioxide are being used to go into photosynthesis, and out comes glucose and oxygen. And so this is correct, right? B is correct. Photosynthesis releases the energy that's stored during the processes of cellular respiration. Cellular respiration is not used to store energy. Cellular respiration is used to release the energy that we put into glucose. Glucose is used during cellular respiration to produce food that is then broken down during photosynthesis. It's all wrong. And again, I didn't change those answers at all. If you understood photosynthesis and respiration, there's only one possible answer here. All right. The potted plant is placed inside a clear sealed vacuum container that has water pumped into it on a regular basis. The container is placed in the sun and the plant's growth is monitored. Which statement best describes what will most likely happen to the plant over time? 2 key points. Again, I almost don't want to explain it because I already explained it in the previous video. I'd like to just reference you to that, but I'll go ahead and explain it here. It says sealed in a vacuum container. So that suggests that there's no oxygen, there's no exchange with the atmosphere. So it's a closed system. All right. There's water. The only thing that's being pumped in there is water. We have to assume that there's no gases in that water. So the plant will grow faster because it's sheltered from the wind. Well, that doesn't make any sense at all. So I don't know why anyone would choose A. Except maybe that you're thinking of some kind of storm where plants are hurt. That's not the point of this. It's not the context. If you look at the context of the question, it doesn't make sense to choose A. It's certainly not the best answer. The plant will die because it receives too much oxygen for photosynthesis. Okay, again, oxygen is produced for photosynthesis. It's not necessary for photosynthesis. Photorespiration would be an issue, and we've talked about that. But this is in a vacuum. It's not receiving any gases, so it doesn't receive any gases, including oxygen. The plant will die because it does not have carbon dioxide for photosynthesis, and this is the best answer, right? It is not receiving gases because it is in a vacuum. And so therefore it's not receiving carbon dioxide necessary for photosynthesis. That's the best answer. The next, letter D, the plant will grow at a slower rate because it requires more water. Water is being pumped into it continuously. So that can't be the situation. That doesn't answer it because the situation is telling you that water is being pumped into continuously. The only reasonable answer is C. And again, this is not changed. I didn't change this question at all from the last time. So I don't know why people didn't get this right. And I explained it in class and then again in the video. You must not be working. I don't know what else to say. Or maybe if it's that you don't understand, you should have come to tutoring. In which of the following situations would you least expect to find anaerobic respiration occurring? This is a new question, but it's basically asking the same content. It's asking about anaerobic respiration. Anaerobic, so here's that word and the prefix "an", which means what? Not. Anaerobic means the use of oxygen, so anaerobic means not using oxygen. All right, so a vat of beer is, and it says, in which of the following would you least expect to see anaerobic respiration? So where would you expect that something's not getting enough oxygen? Well, in a vat in which beer is being manufactured. Well, why would they mention that? Well, they're asking you, do you understand that fermentation, alcohol fermentation is what yeast and bacteria do? We covered that in class. Remember, fermentation is anaerobic respiration. So in humans, we produce lactic acid yeast and bacteria produce alcohol. That's called, fermentation of hops is the making of beer. So this is not a correct answer, because you would certainly expect it. Yes, you would expect it. Right. So we're looking for the least expected. A human brain engaged in writing a test. Well, it certainly takes a lot out of you to write a test, and as you can see from some of the mistakes I made, it's not always easy. But have you ever sat down to read a book or write a paper, and have you ever gotten out of breath by doing that? It's never happened to me, and I don't know of anyone who's happened to that doesn't have some underlying disease. So when you're using your brain, you generally don't run out of oxygen. You're generally not doing it anaerobically. So this is not an issue. This is not an answer. Oh, I'm sorry. That is the answer, because the question is least. So when we're talking about the least expected, I would not expect it to happen here. So I would have chosen B. It's the best answer. But let's go on and look at the others just in case. Inside a bacteria living inside a human intestine. I think to myself, how much oxygen is in an intestine full of water and poop and food, undigested food? I would say not a lot. So I might expect there to be some kind of an anaerobic respiration happening inside an intestine. A runner's leg muscle during a 400-meter dash. 400 meters is a long time. I know that even the best athletes, when they're pushing it to try to go as fast as they can, they're going to be out of breath after a 400-meter dash. They're doing anaerobic respiration in those muscles. So I'm going to go ahead and say I would expect it to happen there. And sediments at the bottom of a pond. Bottom of a pond. It's sediments, so the mud in the bottom of the pond. How much oxygen do you think that exists there? Not a lot. So I would expect there to be anaerobic respiration. So the answer is B. The answer is B. All right, let's move on to 9. What molecule will form when hydrogen ions flow through electrochemical gradient through ATP synthase complexes in the mitochondria? ATP synthase. ATP synthase. Right? This is just simply testing. Did you remember what electron transport chain is? The 3 big things about the electron transport chain. Do you remember it? It's not asking about the cytochromes. It's not asking about the, do you remember the electrochemical gradient means the hydrogen ion concentration gradient, right? ATP synthase. I showed you a movie showing you how it works. I explained it. I drew it on the board. We went through it in class. You took notes on it. I sent you videos on it. You should know what ATP synthase is. So what do you, and even if you don't remember any of that, ATP synthase. You've heard of synthase before, synthesis, to make something new, ATP synthase. What is this enzyme doing? It's making ATP. All right, it's making ATP. Yes, water's produced as well on the side, but not by the ATP synthase. All right, 20. In which way are photosynthesis and cellular respiration different? Here we go again. It's basically, they're opposite of each other, right? So cellular respiration stores ATP. Well, while photosynthesis releases ATP. That's not, no. We know that we're making ATP in order to release it so the body can use it in cellular respiration. And photosynthesis is taking ATP and putting it in the energy into sugar. So it's not A. Cellular respiration produces oxygen while photosynthesis uses oxygen. It's exactly the same answer choice as the previous questions. Over and over again, you're asked the same thing. So if you're not getting these right, you don't know photosynthesis and you don't know cellular respiration. The basics of it, not the complicated stuff, the basics of it, you're not getting it. You need to come to tutor. You need to understand. You need to memorize. You need to study blue cards that I made for you. You need to read the book that I've assigned to you, that I told you read it if you need to. You need to come to tutoring. You need something. Photosynthesis releases energy while cellular respiration stores energy. We know that's not true. Photosynthesis uses carbon dioxide while cellular respiration produces carbon dioxide. That's absolutely true. Remember, I said it over and over again, the two are opposite of one another. The part of photosynthesis that captures CO2 is known as capturing CO2. It's the Calvin cycle. And that's Rubisco in the Calvin cycle. Photosystem 2 and photosystem 1, they're releasing the electrons from water. They're breaking water up. They're producing oxygen. They're making NADPH. They're making ATP, right? That's what the photosystems are doing. Calvin cycle is capturing the CO2 and putting that ATP into this, combining it with CO2 to make glucose, and of course those electrons. The Krebs cycle is in cellular respiration. It's not in photosynthesis. So if you chose C, you don't understand the two processes. All right, move on. Where is oxygen produced in photosynthesis? Oxygen produced. Exactly in the first step. And this is where it gets confusing. It would be photosystem 2. That's where oxygen is produced. It's the very first step. And this was a trick in a way. I warned you about it over and over again. Be careful with prefixes. Light dependent, light independent. The only difference is the IN. The IN means not. So it's not these light independent reactions. The Calvin cycle, and therefore it's not B and C. And notice that the only thing I did to this, the only change I made to this question from the previous exam that I went over with you, is that I changed light dependent to light independent. Light independent is the opposite of light dependent. So C can't be an answer, right? Where previously it was both B and C, now it's just B. All right. 23. Where is sugar made in photosynthesis? In the Calvin cycle. Now here it's not in the, oh look at this, I used the wrong color. It is in the, where is sugar made? It's in the Calvin cycle. I just said it, right? I just explained it. And you need to know it. And I don't know what else to say. But it's not in photosystem 1 and 2, because that's only where you're making your ATP and making your NADPH and releasing electrons and breaking down water and oxygen. And those two photosystems are the light reactions, and it's not B and C. It says light reactions, not light independent reactions, right? Light independent reactions are the Calvin cycle. The English language is tricky. Question 24, what fixes carbon in photosynthesis? Fixes, so what captures, what takes in the carbon during the Calvin cycle? The answer is Rubisco, really the most important enzyme, arguably the most important enzyme on the planet, right? Atp Synthase makes ATP, we went through that. This one's completely made up, didn't change it from the last one. The light reactions, we already talked about what they do, and it can't be B and C, because C is made up. Rubisco is the answer. And by the way, that answer did not change from the previous exam, right? And I explained that question in the previous video. This should be 26, right? One of those mistakes. Maybe I am running out of oxygen. I'm kidding. You know what? Let me correct this. Oh well, it's staying there. Says 26. In which of the following forms is energy immediately made available for use to living cells? There's only one form of these. These are all forms of energy, right? And we've talked about them over in many ways, many times. Let's go through them. Starch, storage form of energy for plants, long-term storage actually in the roots. That's why potatoes are starchy. We've talked about that. So storage versus immediately made available. Well, those two things are exactly the opposite, so it's not starch. Fat is for long-term storage in animals, right, and plants, and so fats are not very, and also for cellular membranes and such, but they're not used for immediate release of energy. Again, you have to take both starch, fats, and glucose. You have to take them all through respiration, cellular respiration, in order to get the energy out. And what form does that energy come out in cellular respiration? It comes out in the form of ATP. ADP is what is the result after the energy is released. It is not the form that holds the most amount of energy. There is some energy in ADP, but ATP is the most amount of energy. So ATP is the answer. It's the best answer. And I'm not going to read question 26. It's basically giving you the answers, walking you through it. It just, either you knew it or you didn't, but this was all just about reminding you about the different kinds of photosynthesis that are happening and why they happen. It was in that photosynthesis video. We discussed it in class that there's these different kinds of photosynthesis that are occurring. There's two different kinds. There's C4 photosynthesis and CAM photosynthesis. There's actually three. There's C3. The C3 is what happens normally, and there's no issues. The Calvin cycle, the light cycles, photosystem 2 and 1, and you produce ATP and NADPH, and the photosystems by breaking down water into oxygen gas and hydrogen ions, and the light photons do that, and the ATP and NADPH move over to the Calvin cycle where Rubisco takes in CO2 and makes glucose, right? So that's photosynthesis. That's C3 photosynthesis. That's normal. That's not what they're asking you about. The question is asking you when you have there's an issue with photorespiration where oxygen gas is taken in instead of CO2. That's a problem. So some plants in places where it's hot and humid, what they do is they change the place of photosynthesis. So instead of it happening where the oxygen gases are where there's a lot of oxygen gas, they move it to the internal parts of their body. Of the internal parts of the organism, so that there's less oxygen available, and so we call that C4 photosynthesis. But that's not what they're asking you, right? They're asking you when you have really hot environments and dry. Not only do you have a problem with photorespiration, but then you're losing water on top of that. So desert plants had to evolve to be able to survive. So cactus had to do something even more special. They had to open their stomata, the openings. Remember we talked about those stomata. They had to open them only at night. So they changed when they do photosynthesis in the different parts of photosynthesis. And so that's the answer. It's CAM photosynthesis for desert plants, and none of the above is not an answer either. So that's the answers. That's the test. You'll see that it's perfectly fair. There may be 3 questions that I would say, wow, where'd they come from? But those 3 questions did come from the notes. They did test the material. They just tested it in a slightly different way. All the rest of the questions were exactly the same from the old exam. I went through them in the old exam with you in class. You took notes on it, theoretically. I didn't see the notes, though, when I graded notebooks. And a lot of your notebooks were missing those notes. And I sent you a video on that as well. So I don't know, guys. I don't know where we're going with this, but if you guys are not working hard on trying to understand what's going on and applying the basics, it's not about memorizing, and it's not a word search problem anymore. We're in a crossword area, right? You have to think about what solving problems using the knowledge you have, the basic knowledge you have, but you have to be able to solve the problem in context, focusing on what question they're asking you. All right, so I hope this helped, and come and see me for tutoring. Identify, you have your answers from your exam. Identify where you got it wrong. Come and see me after class if you don't understand something. Don't get it wrong again. You're going to see the same or very similar 26 questions. Obviously, I'm going to change it up again on the final, all right, as well as translation, transcription, DNA replication, mitosis, meiosis, right? All right, good luck.